This is work in the back. Can you hear? Good. Good morning.
Good morning, sir.
I'm glad you're awake. Better than me. So, as Professor Shoka said, one more day to go besides today. So, in the next hour or so, I'll tell you a story about RNA processing, something else we work on in my lab. So I'm sort of an expert on it, if you will. So this is something probably new to you, right? RNA processing. But you know about eukaryotes, right? Oh, of course you don't. So you all know about transcription, converting DNA genome into messenger RNAs. So in prokaryotes, it transcribes and then that's it. In eukaryotes, the RNA precursors, so-called pre-mRNAs, had to undergo extensive processing. So the DNA genome right here has a lot of introns in them. So the introns need to be spliced out. So splicing happens. So here's the DNA template. Transcription happens. You get the primary transcript, the pre-mRNA. And then the primary transcript has to go processing, including putting a cap at the 5' end, which is very important for the RNA, the messenger RNA. And then the introns have to be spliced out. And then at the 3' end of the RNA has to be cleaved at a specific location, followed by the addition of a poly A tail, so-called 3' end cleavage and polyadenylation. Okay. And then you get the mature mRNA that can be exported from the nucleus. So this is the nucleus into the cytoplasm where it is translated. So without any of these processing events, the RNA is trapped. It doesn't get exported. Then it's useless because it can never be translated. So it's a critical process. So we have been studying the 3' end processing event. We also studied a little bit about 5' capping, but I won't go there. Don't worry about that for now. So if you think about 3' processing, you need cleavage and polyadenylation. Basically, if you think about enzymes, two biochemical activities. You need to cleave the RNA in the endonucleus, right? And then you need a polymerase to add a poly A tail. So two enzymes, endonuclease and a polymerase. So nature does this using a 1.6 megadalton machinery. Very, very complicated. So each bubble here is one protein. And the number you see on that bubble is the molecular weight. So this is CPSF 160, means 160 kilodalton subunit of something called CPSF. But obviously, most of these factors were discovered using classical biochemistry over the last 20 years ago or so. Purified from thymus, all that stuff. I mean, really classical biochemistry. So based on that, they realized that this machinery, all of these 20 or so protein factors form subcomplexes. And that's what these various factors are called. CPSF means cleavage and polyadenylation specificity factor. Again, just like yesterday, the alphabet soup again. Don't blame me, I didn't name them. So that's what we are. That's, I guess, biology, right? That is field you study, there are these acronyms. So hopefully you get familiar. So CPSF, you can see, is involving cleavage and polyadenylation. But more recently, over the last 5-6 years or so, it was realized CPSF actually is made of two subfactors. So that's shown here. MPSF in mammalian systems. So M means mammalian polyadenylation specificity factor, PSF. That's shown in red on this schematic. So it contains 4 subunits, CPSF 160, CPSF 30, WDR 33, and FIP1. So 4 subunits here. And then the second factor is of CPSF, mammalian cleavage factor. As the name suggests, it does the cleavage. So that cleaves the RNA. So that's 3 subunits in pink, CPS 23, CPS 100, and simplicon. And then there are additional factors, which I won't have time to talk about today, so you don't have to worry too much about it. So there's a blue for CSTF, and then CF1M in yellow, CF2M in green, and then RBBP6, another protein down here. So as I said, there are two enzymatic activities. So we need a polymerase to add a poly A tail, and that's known for a long time. That polymerase is poly A polymerase, of course. So that's PAP. So that has a poly A tail. And then the endonuclease that cleave the RNA back when we first started looking at this system was unknown. Even though all of the factors most of these factors were identified at the time, this was early 2000s. But it was not known exactly which one of these is actually the endonuclease that cleave the RNA. The suspicion was CPL73. So that's sort of how it got started. But more importantly, that's also how, now of course we know it is, so that's why CPL73 is part of the cleavage factor, is sitting right next to the cleavage receptors right here. So let me back up a little bit more. So here's the DNA template. Here's RNA polymerase II, transcribe the DNA into pre-mRNA, which is the big black thing here. Here's the stop codon of the mRNA. So this is a 3' UTR, untranslated region, okay, typically. And then how is this cleavage defined? So there are sequence elements within the RNA that helps the machinery decide where to cut the RNA. The most critical one is this hexamer, AUAA, so-called polyadenylation signal, PAS. So this hexamer, AUAA, helps define where the cleavage happens, usually 10 to 30 nucleotides upstream of the cleavage site. There's also some sequence elements downstream that's recognized by CSTF, but as I said, I won't talk about that. Okay, so all of these are good. So as I mentioned, so this was probably 2002, 2004, somewhere around there, let's say. So the idea is CPL73 was suspected to be the endonuclease for the cleavage reaction, but there was no direct biochemical evidence or no evidence to really show that it could be a nucleus. So being a structural biologist, so our goal at the time was to determine the crystal structure of this enzyme. And after a long effort, we were able to determine that structure, which was published in 2006. The structure of human CPL73, the catalytic segment of it, which you can see contains two domains, one cyan and pink, and one yellow. And then even in the back, hopefully you can see there are two little gray spheres here, which are two metal ions, because this enzyme depends on metal ions, zinc, iron, manganese, other things. So these are two metal ions that are important for the catalysis, which I'll show you the mechanism in a little bit. And then also, we see in our crystal structure, there's a sulfate. Hopefully you can see that in the stick model. That's directly coordinated to the metal ions. So we think, based on this structure, that this is the active enzyme. So this provides the first structural evidence that CPL73 can function as an enzyme. In addition, this sulfate could be a mimic for the sisylphosphate or the RNA backbone. So you could imagine an RNA structure binds so that the phosphate RNA binds right here. That's how you do catalysis. Yeah, that's how you cleave the RNA. I'll show you how that's done. So this is sort of how, based on the original structure that we did now nearly 20 years ago now, that's sort of our idea. Okay? However, we also know, as I said, this is a two-domain enzyme, and two domains are closed. They touch each other. So there is no way for the RNA to go into the active site to access the active site. So this is the closed state of the enzyme, and it's inactive. So it cannot be active because there's no way for the substrate to get there. So somehow it had to open up during catalysis. How does it open up? We had no idea. We had one structure back then. So as you see, now we know, again, 14 years later, we know how it works for one system. Okay? So that's sort of the first structure we did. And we did other structures along the way, again, divide and conquer, because something this big, there's no way you can do by crystallography. We're trying to do this by cryo-EM now. We have everything purified. We can see activity of the machinery. But when you put it on the cryo-EM grid and look at the structure, it's too dynamic, very flexible. So it's very difficult to study. So we've been struggling with that for 5 years now. So maybe another 10 years we'll get there. Well, I'll retire by then anyway. Okay, but coming back to divide and conquer. So cyclocan is part of the mammalian cleavage factor, MCF. And now you can see formed the complex with a protein called SH72, which is POL2-CTD. Do you know about POL2-CTD? Well, anyway, POL2 contains many, many protein subunits. And the largest subunit has a long C-terminal extension or C-terminal domain, CTD, that has peptide repeats. So each little bubble is a repeat of 7 amino acids, consensus repeat. And many of them have either tyrosine, serine, or threonine. So you can imagine they're all hydroxyls. They all can be phosphorylated. So the phosphorylation and dephosphorylation of the CTD controls the function of POL2. So SU72 is a CTD dephosphatase that removes the phosphate on the CTD. And then it is shown that this SU72 can form a complex with a cyclocan N-terminal domain. So that's why we determine the structure of cyclocan N-terminal domain, which is in cyan, and the SU72, which is in yellow, and then the CTD peptide, which is shown in green. So it's a ternary complex. So there was a little bit of surprises from that, which I won't talk to. I mean, the paper is here, so you can look at it if you're interested. But anyway, the reason I showed you these structures because I'll come back to them. And then we have other structures we have done, but I won't mention them because I won't come back to them again. Okay, so that's sort of set the stage. So you may wonder, as I said, this poly A signal pass hexanucleotide is critical for this processing event. So then the question is, how is poly A signal recognized by this machinery? And as you know, the poly A signal is recognized by MPSF, which is part shown in red. It's specifically CPS160, WR33, and CPS30 has a critical role in that recognition. So now, this is fast forward many years. So that's the first cryo-EM structure we did in my lab back in 2017. So we determined the structure of the MPSF, which is CPSF160, WR33, CPS30, bound to a hexanucleotide right here, the PAS RNA. So CPS160 in terms of domain architectures has 3 beta propellers. Remember beta propellers? Beta, 4-stranded beta sheet winding around the propeller. So there are 3 beta propellers, so color-coded the same way here. And then WR33 is almost the same size as CPS160. It has a WD40 domain right here. You remember WD40 that forms the beta propeller? So this also tells you that WD40 proteins form a beta propeller, but not all beta propeller proteins are WD40 proteins because these 3 are not WD40 proteins, but they also form beta propellers. Okay, so beta propeller protein has a much larger collection of proteins than just WD40 proteins. But anyway, 3 domains here and a C-terminal domain right here, that's in the front. WR33 actually most of the C-terminal part is highly disordered, so we don't really know what it does. We actually chopped it out. So only the internal piece is shown right here. And then CPS160 contains 5 zinc fingers. So each one of these green bars is a zinc finger. Okay, plus another zinc domain in the C-terminus. But when you see the first 3 in our structure, so you can see essentially you have CPS160, WR33, CPS30 in green, and then RNA is bound between WR33 and CPS30. Okay, so that's sort of shown better on the movie on the right. So here is a more detailed interaction between CPS160 and WR33, which I won't say too much about. And then CPS30 on the right, turn around to the front, and then zoom in. You see 1 zinc, 2nd zinc, 3rd zinc. So 3 zinc fingers. Zinc finger 2 and 3 are important for binding RNA. Zinc finger 1 right here is important for interaction with CPS160. Okay, so essentially that's why only the first 3 zinc fingers are observed in our structure. The next 2 zinc fingers, the rest of CPS30 are disordered. We don't see them. Okay. So essentially, and now you can see the RNAs right here stuck between CPS30 and WR33. CPS160, despite being a gigantic protein right here, has no interaction with RNA directly. So it's a scaffold. So what it does is binds WR33, binds CPS30, so that they are pre-organized to bind the RNA. So that's sort of the idea. All right. So how does it bind? So it's A, U, A. Are you familiar with looking at nucleotides? Protein is probably more than nucleotides. But anyway, here, I got used to myself. At first, when I started doing this, I need to get used to looking at nucleotides. But anyway, so even though it's a little hexanucleotide, actually it's quite busy. So A1, A2, right here, you can see the base. A1 base is here, A2 base is here, pointing into the screen, right? And they interact with zinc finger 2 of CPS30. And then A4, A5 are in the plane. They point up, and they interact with zinc finger 3. And then you can see the A's on the top of protein kinases. I'll see more. You know about AT base, Watson-Crick base pair, right? AU base pair, AT base pair. They are formed by using N1 and N6 atoms of the adenine. You can see in this case A4, A1, and N1 and N6 both are directly hydrogen-bonded to the zinc finger of CPS30. So you can imagine that that's how you recognize the A in the DNA duplex or RNA duplex. This is how the protein recognizes the A here. And then A5 forms only 100 bond to the N1. N6 is not involved. Same is true for A1, A2. So essentially, these adenines are recognized directly by the zinc fingers. Now, on the other side, U3 and A6 form something called a Hookstein base pair. So now the direct Watson-Crick base pair was side-to-side base pair, which is a complete surprise. I mean, we didn't expect that something, a little hexanucleotide, can form a Hookstein base pair between the third and the sixth bases of the nucleotide. So you can see the backbone is running back and forth like a snake, the X-shaped structure. The other thing we see is the binding mode of the two nucleotides to zinc finger 2 and the A4, A5 to zinc finger 3 actually quite similar. So if you overlay the two zinc fingers, zinc finger 2 and zinc finger 3, the nucleotides also overlay. Essentially, the nucleotide has the same binding mode to zinc fingers. So the idea that essentially the zinc fingers have a conserved mechanism recognizing an AA dinucleotide, or prefer than AA dinucleotide, because as I showed you the 100 bonding scheme here. Okay? So that's good. So as I mentioned, we have been trying to study the entire machinery now by using cryo-EM for quite a few years now. Not just us, many other labs around the world are doing the same thing. And because the system is so dynamic, it has been very difficult to observe them in a stable conformation that we can see how all the subunits are coming together. So as I said yesterday, you cannot make progress on something, you go sideways, look at something similar. So in this case, so this machinery, I forgot to mention at the beginning, is what I call the canonical machinery, because this is used for 3' processing of the vast majority of mRNAs in eukaryote or in animals or mammals. But in us, in humans and animals, for replication-dependent histone pre-mRNAs, that process using a different machinery. That's the one on the right. It's called the U7 SNRP. So it's a SNRP. And this one's 1 megadalton, so not much smaller. You can see it's entirely different between the two. The overall protein composition, everything else, very, very different. I'll say more about this on the next slide, but what I want to highlight is here, the one that does the cleave the RNA is this factor here called HCC. Sorry, another acronym, histone pre-RNA cleavage complex. And this HCC contains CPS73, CPS100, supplicant. You may remember that's exactly what's in MCF, CPS73, CPS100, supplicant. So even though the two machinery have very different composition into the other protein factors, they share the same cleavage module. So nature reused the same module in two different cleavage machineries. Okay. So my collaborators, we met at a meeting in Japan, in Kyoto. They really twisted my arm during the entire meeting, say, Leon, you got to work on this, you got to work on this, you got to work on this. I said, okay, I'll give it a try. So the rest is history, as you say. So what is U7 SNRP? So first of all, it's a SNRP. So that's a USAM ring. So made of SM proteins. SM is a protein motif. That each one has this SM motif that form this ring, 7-member ring. You all know about splicing, right? So splicing also contains these rings. Spliceosome has SNRPs in there. It also contains the ring. The difference is the spliceosome rings have different subunits here. They share B, D3, G, E, and F subunits. But LS10, LS11 are unique to USAM SNRP. That's why it's a USAM SNRP, because these are two unique protein factors in this ring. And the ring recognizes the RNA, the USAM siRNA, which is right here, 5' here, 3' down there. That's what the ring does. So there's a SM motif that threads in the middle, the central hole of the ring. That's how it's recognized. I won't have time to show you that today. So that's the USAM siRNA and the SM ring. And then the pre-mRNA is this molecule right here, shown in orange. And they contain a 5' conserved stem loop structure right here. And that's recognized by something called the SLBP, stem loop binding protein. More importantly, downstream of the cleavage site, there's something called histone downstream element that forms the duplex with the 5' and the U7 siRNA, which I will refer to as the HTE U7 duplex to make it easier to say. Okay. And then, as I mentioned, LS10, LS11 are unique to the U7 SNRP. Especially LS11 is a very large protein. Most of these proteins only about 100 amino acids. LS10 has 360, a very large protein. So the first 150 residues or so are outside so it's an internal extension. That's shown right here. So the SM motif is these two purple things. So there's a large insertion in the middle of the SM motif, which sort of comes out of the ring somewhere. And then there's a very long internal extension. And one important function of this extension is to interact with a protein called FLASH. FLASH, okay, I'll come back to FLASH a little bit. And then LS10 is, as you can see, 120 residue, very small protein, has a SM motif, and then a small N-terminal, small C-terminal extensions. So as I mentioned, LS11 has very long N-terminal extension, so that drew a lot of attention. So people studied it. They chop it out. They realized that if you chop it out, the machinery is dead. So it's critical for the functional machinery. LS10 has small extensions, even though it's unique to the U7 SNRP, because these extensions are so small, people basically ignored them, didn't really pay attention to them. Nobody really studied them. So we'll come back to that. So as I mentioned, LS11 internal extension recruits a protein called, or interacts with a protein called FLASH. FLASH is a gigantic protein, 2,000 residues. Okay. And then FLASH in turn recruits ACC. So this is how the whole machinery works. You can say the U7 ring forms, LS11 reach out, recruit FLASH. FLASH in turn recruits ACC, and that RNA gets formed in the duplex, and the whole thing ensembles. Okay? Does all make sense? All right. So that's sort of the idea. And HTC is the same MCF, you know. So we start, as I said, we met the collaborators at a meeting in Kyoto. So we started probably 2012. And then the first structure we did is how this conserved stem loop is recognized by SLBP. That's shown right here. So here the stem loop structure, stem, a loop on top. And the SLBP is on the right. It's only the RNA binding domain SLBP is shown right here. Especially this helix here measures how long the stem is. So these are very important interactions. There's another enzyme that we use to form a ternary complex, but I won't have time to talk about it today. So that sort of gives us idea how this part works. And the next thing is FLASH, I just said, is a 2,000 residue protein. But most of that is disordered. Only a very small piece of it is required for histone PRMA 3' processing, based, again, through the work of our collaborators. Zbigniew Dominski and Bill Marcelov in Chapel Hill, North Carolina. So we then determine the crystal structure of that piece of FLASH that's required for 3' processing. As you can see, it's only about 60, 70, 80 residues of FLASH. And to our surprise, that piece forms a coil-coil dimer. Remember coils that I mentioned on Tuesday, I think? So essentially, rather than having only one of these gigantic proteins, you need two. That's why this is the 1 megadalton machinery, because two of these are already 450 kilodalton. So half that 1 megadalton came from this single protein. But still, we'll take it. So what we know based on biochemistry, the C-terminal part of this coil-coil dimer is where LS11 interact with. So this interaction stabilizes the flash LS11 complex. And then N-terminal just beyond this coil-coil, 20 residues, is a part that interacts with HCC. As you see later, actually interact with symplecin. So this piece. So flash is the bridge between LS11 or the ring and the HCC. Okay. So now we have pieces of this by dividing and conquering. So then the question is, how does this machinery work? So that's usually the case. We try to do everything recombinant. So we try to reconstitute the entire machinery. So we have SLBP from this. We have HCC or MCF from our studies of the canonical machinery. So we have that protein expressed and purified. We have flash right here. So the first thing we try to do now is reconstitute the entire ring. And then after a lot of effort, my postdoc actually succeeded in making the ring. So we have everything together. So now we can put them together, mix with the RNA, and see whether we can see cleavage right here, right? To see whether we get activity using this totally recombinant, reconstituted machinery. So that's what we did. And then the first sample we used for that, for HCC, we chopped out the N-terminal domain of symplecin. Because as I showed you at the very beginning, this domain forms the complex with SU72. So we thought we understood what this domain does. We don't really need it. It interacts with SU72, probably is not required for the function. So we chopped it out. More importantly, if you chop this out, the remaining sample is easier to make. You can make morbid, purer, and everything. So that's why we did it that way. However, so this is a model histone pre-RNA substrate, radio labeled. So this is work done by our collaborators again. So this is the control experiment, just RNA alone, no reaction, nothing. And then we mix everything, all the material for the machinery, together with HCC lacking the NTD. We don't see any cleavage reaction. So enzyme is dead, doesn't work. So I thought, okay, what's missing? Looking through everything, I feel the biggest piece that's missing is the NTD of symplecin. But as I showed you, we can purify that as a protein on its own. So we purify that protein, send it to our collaborator. They add it in trans as a separate protein. And lo and behold, when you add it in, we got beautiful cleavage activity. So RNA got cleaved to a smaller species. This is exactly what you get using nuclear extracts. So this is the genuine product that's formed by adding NTD as a separate protein. So that shows the NTD actually is very important. But also, actually, we also did gel filtration experiment to show whether the NTD can form a stable complex with the rest of the machinery. It does not form a complex. It runs on its own. So even though it's critical for the reaction, but it does not stay together with the complex. So now that we understand the NTD is required, we don't have a choice. Even though it's harder to express, we have to go back, make the full length symplecin. So now when you have the full length symplecin, you don't need the NTD anymore. So you get activity with or without additional NTD. It doesn't matter anymore because there's NTD already in the full length protein. And then we make a mutation in the anti-cytosine CBS73, you knock out that cleavage activity. So this is actually the first time that's direct biochemical evidence showing that recombinant CBS73 can cleave RNA, and then if you mutate that site, you knock it out altogether. Now I showed you the NTD binds SU72, and it turns out that in reaction that use NTD as a separate protein, this reaction right here, SU72 actually is an inhibitor of the reaction. It actually inhibits pretty potently. And from our studies back then, we know if you make a mutation in the NTD of symplecin, a single site mutation, you block binding of SU72. And if you use that mutant, it's no longer sensitive to inhibition by SU72. So everything seemed to make sense. biochemically. So all of these together shows that the NTD of silicon is required for processing of the histone pre-RNA, histone pre-RNA. So it's a required factor in the U7 nerve. So this is all good. The ultimate goal, of course, how does it work? So now we have all the proteins. We mix them all with the 2 RNA molecules, essentially. So we have 13 recombinant proteins and 2 RNAs ordered from IDT, synthetic, and then run them on gel filtration. We've got 2 peaks. Peak 1, you can see if you count it out, contain all 13 proteins, 13 bands there. So silicon, CPS100, CPS23, and then CSCS64, LS11, LSLBP, flash right here, and the rest of the ring. They're all there. Peak 2, you can see, contain primarily the ring, no HCC. And then the 260 is red. It's higher than 280, means there's nucleic acid there. That's because there are 2 RNAs bound to the machinery. So this is a sample that we purified and put on the EM grid, froze it down. That led to a structure of the overall core of the enzyme, published now 5 years ago now at 3.2 ounce resolution. So here, down here, the U7 ring. So 7 subunits, 7 different colors. And then over here, you can see the duplex. And then here is CPSF73, brighter colors, CPS100, a little bit darker colors. And then over here is the silicon NTD. Remember, that's required for activity. That's right here. So here the duplex again. Zooming in, so the NTD is required, is involving recognizing the duplex. Together is 100 CPS73. Here's a little piece of the internal extension of LSI11. Most of it we don't see. So this is just a very small piece of it. And unexpectedly, we actually trapped the RNA in the active site of CPS73. So this is the active site, that's the 2 metal ions. Okay, should I let it go around one more time? Okay, so I guess Professor Shukla mentioned writing a paper. Writing a structural paper, first thing you decide is the color scheme for your structure, because it will be important to keep the same colors throughout the paper, because otherwise it'll be very confusing. And so something with 13 proteins, you run out of colors pretty quickly. So you have to be creative of making colors. And also, some of these actually are colors from way back from the first paper in 2006. I chose the color scheme back then already. I try to keep the same color scheme here as well. As you see when I zoom out, so cyan and yellow. Remember the first shot I showed you was cyan yellow? Cyan yellow. So I kept the same color scheme. Okay, this is something that when you write a paper to tell a story, make it consistent. Okay? Anyway, so this is sort of the overall structure of the machinery, which I call the core, because as you saw, we have flash in our sample. But there is no flash here. Flash the coil diameter. You don't see that at all. Just because this is the core. The whole thing looks like this. Flash is right here. And then this is silicon CTD. So flash talks to the CTD of silicon. And then the whole thing looks like a Greek Asian amphora. But this one has only one handle, but that's easy to do. It's one handle. So flash will be the handle, and then CTD is another part of the handle, and the core of the machinery is the body of the amphora, and then at the side right in the middle. And then finally, the stem loop structure is right here. Even though it's highly conserved, also in the periphery. And the periphery is much more flexible. That's why we get a much lower resolution structure of it. So, but the core is where the biochemistry is done. So that's the most important part of it. So I can say a lot more about this, but the main thing I want to say today is how the RNA is cleaved by this machinery. So as I said, we did not expect this, but we somehow trapped the RNA in the active site. So this you can see is the open state. The 2 domains opened up. The RNA is bound into a deep canyon between the 2 domains. In comparison, our crystal structure earlier of CPS23 alone, in the closed state, you can see there is no canyon, as I mentioned. There is no way for the RNA to go in. So here RNA is bound. This is the cystophosphate right here. That's coordinated to metal ions, and that's shown right here. So here the cystophosphate, and then in the thinner sticks is the sulfate that we saw earlier in our crystal structure. It binds the same way, just as we suspected back then. So this sulfate or the phosphate directly coordinates the 2 metal ions. in the active site. And then the metal ions are surrounded by many side chains, histidine and aspartate. And then bridging the metal ion is a water or hydroxide that sits directly below the phosphorus. So this can initiate a nucleophilic attack on the phosphorus. And this 3' hydroxyl will be the leaving group at the hydroxide oxyanion, which will be very unstable. So that needs to be protonated to stabilize it. And that's done by this histidine, which is activated by this glutamate. So all of these residues are strictly conserved among CPS73 homologs. That tells you essentially that these have important roles in the enzyme. As you can see, they are critical for the catalysis. So the bottom line is the binding mode explains the mechanism, and the CPS73 is now in the active open conformation, and the pre-mRNA actually is poised for the cleavage reaction, ready to be cleaved. And then you can see the binding site is very narrow, so only a single-stranded RNA can go. Double-stranded will not be able to get in. So now how does the enzyme open up? So clamshell model. So if you do an overlay the structures, then it corresponds to a 17-degree rotation of one domain relative to the other. Okay? So this figure, or these figures or movies are made using Pymol. If you don't know how to do it, I'll show you how to do that, how to make a movie like this in Pymol. So very helpful, right, for you to understand the biochemistry or the conformational change in the system. It takes a little bit of effort to make a good movie, but once you make it right, very, very useful, I think. So that's why I keep on showing them. So front view and side view may be better. So now closed state, everything closed down, the RNA cannot go in, open up, RNA can be there. You can make it fancier than what I have here if you have more time, all that stuff. Okay, and more importantly, the question is, how does it open? Or why does it open? So when we look at the structure, around the yellow domain right here, which I won't name it, around the yellow domain, there are two little segments which are colored green here, next to it. Where did they come from? They came from the N and the C stimulus extension of less than 10. So the idea then essentially, the close contact between this domain of CPS73 and these N and C stimulus extension of less than 10 is important for opening up CPS73. That would suggest an important role for these two extensions, which, as I mentioned earlier, nobody cared, nobody studied. So based on that, our collaborators did biochemistry, mutagenesis work. So if you make them wild-type machinery, you get beautiful cleavage reaction. Now, if you delete the internal extension of zone 11, the enzyme is dead. Well, that's known, as I mentioned earlier. People studied it. But then if you delete the C-terminal extension or both extension less than 10, the enzyme actually is much worse. In addition, you can see a second bench starts showing up. So not only the N and C-terminal extension are important for the activity, it's also important for generating the correct product of the RNA. So it's a very important two little pieces of protein for the activity of this machinery. That insight came only from the structural analysis. So as I showed you in the movie already, the NTD of simplecan is help recognizing the duplex. So the idea that essentially simply NTD comes in, recognize duplex, pushing against CPS 100, which in turn pushes against CPS 93, which again push against less than 10. That's how the thing opens up. So that's our model. So this will explain then why the NTD of simplecan is required for that cleavage reaction, because without that, you don't start the triggering of the process. Then I also showed SU72 inhibits this cleavage reaction. It inhibits because it binds at the same place as the duplex. So they are mutually exclusive. If SU72 binds, RNA cannot bind. That's why it inhibits. So very simply explaining that process as well. And there's one more aspect that let me explain a little bit. So here I show you an overlay of two structures. So in the cartoon drawing in color, is the structure we observed in the U7 SNRP in the active state. So CPS 100, CPS 23 in the close contact, and then simplecan up here. And then in gray is EM density envelope displayed by a chimera or whatever. And then you can sort of see now you have CPS 100, CPS 73, but they are opened up. They don't talk to each other. So this is the active state. The opened up state is the inactive state. You can see they're quite different. You can also imagine a movie like this, the difference between the two states. Okay? So this movie took some time to make as well, but anyway. So that's done using chimera, which I'm not very good at. But anyway, so you can sort of clearly see the computational change between the inactive state like this, and then in the active state, the two things come together. So in addition to recognizing the RNA, there's also computational changes in the cleavage factor, or ACC. So putting everything together, another movie. So USN rings formed, and then LSI 11 reaches out and recruits flash dimer, and then flash dimer reach out, recruit HCC. So now you have USN SNRP. Now mRNA comes in, form the duplex right here, HD-U7 duplex, but machine is still not active. Then a subsequent NTD comes in, right on the duplex, large computational changes in the HCC, especially CPS23 opens up, RNA slots in. Now you have everything ready for catalysis. And somehow we trap this state of the enzyme for cryo-EM. That's what we determined. After cleavage, the mature RNA leaves, the downstream RNA gets degraded, actually also by CPS23, and then NTD leaves, the whole thing disassembles, and then you can reassemble for the next RNA molecule. Does that sort of make sense? So that's sort of the way we think about it. So if you think that way, you have a cycle of life, if you will, for this machinery, going from the assembly of the U7 SNRP to recruiting HCC and then RNA and then recycling everything back and forth. So far we have determined the structure of this state of the cycle. We'd like to determine the structures of other states of the cycle, but just like the canonical machinery, these states are highly dynamic. So it'll take some effort for us to understand how to stabilize them so then get them in the right state. So, but at least we have one now that gives us tremendous amount of insight on this machinery. Okay, all right, not too bad. So I already said most of these. So you're interested in drug discovery, I guess, right? So if you think about it, these are critical events in the cell, as the way I explained them. Then you would imagine that CPS23 being the nucleus for both machineries, you can't really touch them. You cannot really touch that CPS23 because if you mess around with CPS23, you probably will kill the cell. However, the same year we published our paper on the U7 SNRP structure, scientists at Novartis in Cambridge, Massachusetts, published a paper showing that CPS3, which is the systematic name for CPS23, so same protein, two different names. And they showed that CPS23 actually could be a druggable node against cancer, AML or urine sarcoma. Completely unexpected. So where did that story come from? Came from even before that, from 1999. So came from this compound called JTE607. So that was discovered by Japan Tobacco, JT, in 1999 as an anti-inflammatory agent. So I know because back in 1990s, I was working on anti-inflammatories myself. So that's sort of everybody back then was working on anti-inflammatories, which I'll tell you about tomorrow. Okay? So this was found as an anti-inflammatory agent based on cell-based assay. So not purified protein, nothing. You basically cell-based assay. You challenge with something, you look for response. And later, this compound was shown to also kill AML cell lines. Okay, acute myeloid leukemia. But as I said, these are cell-based assays. So the exact target of this compound in the cell was unknown. So what scientists in Novartis did is they first screened the compound against 92 cancer cell lines. And they found that to be active not only against AML, also against urine sarcoma. So 2 cancers now, not just one. More importantly, using modern technologies, siRNA screening and chemical proteomics, they found that the target of this compound is CPS73. So this will be the first known small molecule inhibitor against human CPS73. Even more interestingly, if you look at this compound, on the right, you have an ester, right? So this compound itself is inactive. So it needs to be activated by carboxyl esterase to hydrolyze the ester into an acid. The acid is the active species, the ester is not. So in that sense, the ester being uncharged is very good for the compound to transverse, to go across the membrane. And once in the membrane, it's activated with the enzyme, and now it's active, also it's trapped in the cell. So somehow, scientists in Japan developed a protein We're back. I took a nap. That was good. Okay, so the story is we have the acid being the active species. The ester is not active. So they used the active compound, did the bonding assay, and showed that KD is about 370 nanomolar, which is good, but not great. I mean, you all know for a good compound, it should be down in the nanomolar range, single-digit nanomolar probably, or even below nanomolar. So there's probably still a lot of room to improve in terms of binding affinity between the compound and the target. More importantly, they determine the crystal structure of this compound bound to human CPS23. So they see two zinc ions. In this case, they see a phosphate bound to the zinc ions, the metal ion. So the compound does not talk to the metal ion at all. It binds near the active site next to it, right here. And most importantly, remember the carboxylic acid required for the activity? That's right here. It has direct 100 bonding interaction with the backbone amide of the protein. So very close contact between the acid and the protein. And then over here, just a cavity in the active site region of CPS23 showing that CPS23 showing there's a pretty large cavity. The compound sits in the middle of the cavity. And we will PyMoling as well. We'll PyMol that. We'll show you cavities as well. Very easy to do in PyMol. Okay. So that's all in vitro. In vivo, how does it kill the cell? The idea is CPS23 cleave the primary transcript is important for alanine polymerase termination, transcription termination. So that's in the normal cells, the transcription terminates, everything normal, you get cell growth. But then once you inhibit CPS23 using this compound, you don't cleave the RNA anymore, so POL2 keep on going. So you get transcription read-through. And that leads to all kinds of problems for the cell. And then in the end, what did that do? In the end, you have a defect. That's how the cells die, because of the read-through problem. Essentially, once this cleavage happens, there's another machinery that comes in that chases down POL2 and then evicts it from the DNA template. Without this cleavage, you can't do that anymore. So that's the basic idea. So that's all good. And okay, so I went back and looked at the original 1999 paper from Japan Tobacco, anti-inflammatory in humans, monkey, rabbit, cytokine production, anti-inflammatory, right? So in their hands, JT607 actually is very potent. You can see this response curve. It's about low single-digit nanomolar. So I still don't understand myself how the compound is so potent against cytokine production, but has a KD of 370 against CPS23. So something else may be going on here as well that maybe somebody else will figure it out. Okay? So CPS23 now actually has some interest as a cancer.
Hello, hello.
Nope, doesn't work either. Okay, that's fine. I only have 5 minutes. I'll yield. All right. Boron, that's also boron. So this compound AN3661 is an antimalarial compound. It has very good potency, low nanomolar potency, different malaria isolates. And they also determine the structure of this compound bound to a parasite, CPS-73, not malaria, get results. So this one here, the cryptosporidium, CPS-73. And they found essentially the boronic acid part gets hydrated, is directly chlorinated the zinc lines in the outside, which makes sense. As you can see also, AN3061 has carboxylic acid. And that carboxylic acid is directly around CPS-73, just like carboxylic acid in ATP-citrate. So if we imagine that CPS-73 has a little sweet spot here for binding of carboxylic acid. Both AN3661 and JP607 use that binding site. Okay? That's all good. So because of this interest in cancer, so many groups around the world looking for inhibitors in CPS-73. So a group in China published in 2019, actually, developed this compound, 21 nanomolar against ovarian cancer cells. But they didn't identify the target of this compound itself. So again, sulfate acid. But you could say, the compound looks just like AN3661. So the target should be CPS-73. But then, at the same time, I said, this carboxylic acid directly contacts CPS-73. And this compound has a very large substitute here. There is no room for the compound to bind the same way to CPS-73. So there is no room for the compound to bind to CPS-73. So we still, it's not clear this is the way the compound works by inhibiting CPS-73. So this is where our collaborators came in, Deepak and Jeff at UT Southwestern. So they developed something called the inducible mutagenesis system to identify resistance mutations. The basic concept very simple. You have a cell line where you can turn on mutagenesis, and then you challenge the cell line with this compound. And then for the surviving cells, you do exome sequencing to find which one where are the mutations that allow the cell to grow, confer resistance, right? So the one, hopefully the target is the one that gets mutated the most. So that's what they did, again, using this compound. And what did they find? So they find CPS3, or CPS73. Out of 11 clones that are resistant to this compound, they identified all 11 carry mutations in CPS73. And most interestingly, proline 45, hope you can see in the back, was found 5 separate times, different mutations, proline to leucine and proline to threonine. So they're found independently 5 different times, where other genes are mutated sporadically, so probably they're not really essential. So this suggests that CPS73 is actually the target of this compound. So again, this Deepak and Jeff certainly were not expecting this. They found this, and they again realized I'm the expert on CPS73, so they reached out to us and said, could you do an assay, cleavage assay, to show that this compound does inhibit the cleavage? I said, we can do that, but we can do better. We can get the structure of this compound bound to CPS73. That will be even more interesting or insightful. So that's, well, one more data from them. So they used this compound here and did click chemistry to show that the compound can pull down CPS73. And then when you make a when you include the mutation, proline mutation, tyrosine mutation, you put down much less. Again, showing you specificity. Okay, this again work from Deepak and Jeff. So what we did is they sent us the compound. We determined the crystal structure of human CPS73 bound to compound at 1.700 resolution. You're good at looking at electron density now, I hope. Here's the electron density of the compound, really beautiful electron density, large bump for the carbonyl carbon, oxygen, sorry. And then briefly, just the binding mode of the compound, you can see the benzoyl part directly coordinate the metal line, just like N3661. And then everything else lies along between the two interfaces, the interface between the two domains of CPS73. And interestingly, the residue labeled in red are the ones that are found to confer resistance when mutated, especially proline 45 right here, directly on top of the inhibitor. So that the structure confirms that the mutations either directly affect binding or indirectly in the interface between the two domains, which probably affect the open-close transition that affect the binding of the compound. Now, if you compare the binding mode of our compound, or no, sorry, not our compound, the compound in the literature against JTE607, you can see actually the binding mode is very, very different. Again, showing you the power of structural biology that without that, you would not even know how to orient everything. So the compound 1 has direct contact with metal line, goes around this way. JTE607 actually almost perpendicular to it. Most importantly, you remember this carboxylate is essential for the activity of JTE607, but it's not used as a part of the binding site for compound 1. So if you think about doing structure-based drug design, maybe you can think about adding something here to grow right there to sort of try to reach that binding site for the carboxylate. That would be helpful. Now, if you compare to the binding mode of N3661, same thing. The carboxylate point up in the N3661 interacting with CPS73 directly. Compound 1 has a large group here, so it cannot go there. So the whole thing flips upside down. That's how it binds. So you could sort of say that this is represent a new class of CPS73 inhibitors. And in the interest of time, I will skip over the left side to talk about the transcription read-through. So Jeff and Deepak did an experiment. So did sequencing to show that in the wild-type cells, you can see the transcript goes from right to left. First gene RLP, followed by SCARF1, second gene. You can see in the wild-type cells, if you treat with the compound 1, you get read-through. DMS control, you don't. So compound does promote read-through. And secondly, using a resistant mutation, Y170H, you don't get as much read-through. So it shows specificity that this compound does cause read-through in the transcription, and that's how it probably killed the cells, just like JTE607. And then they also screened against a large collection of different cancer cells from different lineages, and so that this compound actually kills almost all cancers. Okay, in contrast to the JTE607, which only kills AML and urinary sarcoma, because only those two cell lines have the carboxyl esterase that can activate the compound. But this one does not need to be activated, so it's active against a whole series of cancer cell lines. So this was published last year. So last little bit, 2 minutes probably, we tried to do structure-based drug design. So as I mentioned, first idea suggested to Jeff and Deepak was to grow something here to reach this carboxylate. But the chemistry there was intractable, very, very hard. The next thing we tried, we look at the comparison between binding mode of compound 1 and JTE607. This group right here, which seems to come up with this carbon from the compound 1. So we thought we can graft this substituent right here, and then maybe improve the activity of the compound, maybe also the properties, other properties. So they made a compound. It is still active, which is good. Not much more active, but roughly the same activity. And then send us the compound. We determine the structure. That's showing green here. So based on prediction, you expect this ring here to follow JTE607, so go along in the plane of this view here. Turns out in our crystal structure, it points towards you, 90 degrees away. And then when we really look at the structure, we understand why it goes this way. But then when we design the compound, we thought this way would be the right way to go. So again, it shows you that it's a structure-based drug design depends on structures. So you said you can do many, many calculations along the way. You get activity and everything, that's great. But in the end, along the way, once in a while, you need a structure to make sure you're still on sound footing. Otherwise, you could be really off in somewhere you don't even know about. So, last thing, so 2022, just CPS73 is a cancer target. 2022, disease-causing mutations of CPS73 was found in the general population, in children mostly. So this is the first paper, actually quite a few now, papers on this. And then they show that in this case, 6 homozygous mutations of CPS73 show severe intellectual disability, seizures, and microcephaly. So now CPS73 has a role in intellectual disability. How that works, obviously, we have no clue. This is very, very new. So I don't have a slide for that. So actually, Reza Marufi in London contacted me, again, as an expert of CPS73, saying he has a collection of CPS73 mutation himself that's linked to mental intellectual disability. He liked to understand, he is a geneticist, he look at patients, find these mutations. He wants us to help him figure out what the function of those mutations are. So we are trying to do crystal structures of mutations, and also doing assays to see how the mutation affect the cleavage activity, and then maybe beyond that, deep sequencing to see how it affects RNA population, this and that in the cell. So, and then maybe there's also tissue specificity, this and that. So all of these are brand new stuff, very interesting new area that we completely did not expect, because how would you expect CPS73 mutations linked to intellectual disability? But that's science. You go with what you have. So very exciting thing coming on actually in the coming going on in the coming years. So with that, I'll stop. Hopefully you learned something. If there is time, I'll show you a couple of the structures that I talked about this morning. Okay, next, Professor Shukla.
Yes, so probably we can have break for 10 minutes. Okay, we can have tea because 12:30 we will have a final break. So right now the tea and sandwich is there. You can have something, otherwise again some of you may be feeling weakness. Okay, please go and take tea, and then I will join in 10 minutes.